Hey everybody, and welcome back to Introductory Biology. Just a little bit of housekeeping before we get started here. Congratulations, you guys are officially 1/4 of the way through the semester. So we are starting Unit 2, and Unit 2 has to do with cells and cell structure. So let's get started. So this image that you're looking at here is an image of a human egg inside a fallopian tube. And interestingly enough, I am actually a reproductive physiologist, and so this image here is kind of an exciting image for me to look at. Each and every one of us, we actually started out as one cell, and so this is kind of looking at a picture of how we all were at one point in time. So this is what we call a zygote. So a fertilized egg is a zygote. That one cell then divides into 2, 4, 6, 8, 16, 32, and then becomes a ball of cells, and then eventually moves its way down the fallopian tubes and into the uterus, becomes implanted into the uterus, and develops over time into a baby. So kind of cool. So today what we're going to talk about is the cell and the structure of cells and the function of cells. And so let's get started with that. So there's a couple of different things that we need to know before we kind of delve into the complexity of cells and what makes up a cell and the inner workings of a cell. And the kind of the basics of cell is something that we call cell theory. So there's 3 different parts to cell theory. And 1, all organisms are composed of 1 or more cells. 2, a cell is the smallest living, makes up the smallest living things. And 3, cells arise by the division of previously existing cells. So there's only 3 different components to cell theory, so make sure that you write them down or take a look at this slide and commit them to your mind. So Robert Hooke was the first individual to actually discover cells. So, for a long time we didn't even realize that our own bodies, our own skin was made up of cells. We didn't realize that our liver was made up of cells, or our heart was made up of cells, that our eyes were made up of cells. So we had none of that knowledge of cells. What actually brought us to the knowledge of cells was the discovering of a microscope. And so in this image that I have right here is looking at basically 3 different types of microscopes, and you guys will of course have labs on microscopes and stuff, but the first type of microscope that is shown in this basic image right here is something that we call an optical microscope or a light microscope. And what a light microscope does, or an optical light microscope, is that it takes light and lenses, which it uses to actually bend and magnify the image that we're looking at. And there's a couple of different types of light microscopes that I'm sure that you could be able to see in lab and take a look at, hopefully you've looked at them in real life and not just in an online course, but you've been able to actually hands-on work with them. But they're kind of cool to actually magnify different things and take a look at different cells, whether it is a plant cell, or perhaps an onion cell, or blood cells underneath a microscope. And then what we have are a couple of different what we call electron microscopes, and that's these right here. So the 2 different types of electron microscopes, one is what we call a TEM, or transmission electron microscope, and the second one is what we call a scanning electron microscope. So the transmission electron microscope takes magnets, and these little gray things that you see kind of marching down the side here are magnets, and we'll take these really, really high-powered magnets, and we'll take the object that you're interested in looking at, the person that's working with the object will actually take really thin slices of that object, and then you will actually transmit through that object and bounce electrons off of it in order to see the object more clearly. So it's kind of really kind of cool in order to see that image. A scanning electron microscope will actually take the object, and you put a thin layer of gold over the surface of that object. So again, scanning electron microscope, you actually scan over the surface of the object, again, that you've put gold on the top of, and you use these, again, magnets to bounce electrons off the surface of that object. And so you can see really fine detail in both of these electron microscopes. The transmission, of course, transmits through the microscope, it's through the object, and the scanning electron microscope actually looks at the surface structure. So this image that you see right here is actually looking at a cell, and they're using TEM, or transmission electron microscope, microscopy, to actually look at the inner workings of this cell. So that's kind of cool. Well, that's kind of crappy. When I imported this, it kind of shifted these images down, and when it shifted these images down, it covered up these words. But I'm sure that you guys can see the original document, and you know the words here says, "Why are cells so small? Why can't they be as huge as hippos?" And so one of the reasons why cells are so small has all to do with the surface to volume ratio. And if I make a square here, and I talk about volume to surface ratio, cells have to be small in order for oxygen to move. I'm going to try to make an arrow. Oxygen to move into the cell, and then carbon dioxide to move out of that cell. And so if you think about it, if you take this cell right here, and now you divide that cell up into fourths, and so now you have 4 different individual cells that are much smaller. So now let me see if I can draw 4 small boxes that are now cells that are smaller in size. Oxygen and carbon dioxide actually diffuse across the membrane. So now oxygen is going to move into that cell, and carbon dioxide is going to move out of that cell. And this actually has a shorter route to go, if you will. And we have a shorter distance to go. And if you break this larger cell up here into 4, you can have a lot more activity, if you will. So now we've got 4 baby cells, and we have a lot more oxygen and carbon dioxide. Oxygen going in, carbon dioxide going out. Oxygen going in, and carbon dioxide going out. If we take each one of these baby cells here and divide each one of these now into fourths, so again, each one of these little boxes, think of it as a cell, and now we're making each one of these boxes into 4 separate cells. My drawings are getting a little complicated here, but you know what I'm talking about. So now we've got 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16 small little baby cells that I've made. And so I'm not going to draw them all out, but what I'm going to do is draw the very first one. So I'm going to draw this one right here for you and make it a little bit so you understand. So that one that I just put the circle around, now what we've done is we've made that one into much smaller cells. Okay, so now we've taken our surface area and we've made it even smaller. So again, the passageway that oxygen, the distance that oxygen has to travel to get into that cell is much smaller going in and much smaller going out. Much smaller going in, much smaller going out. Much smaller going in, much smaller going out, much smaller going in, and much smaller going out. Again, you've increased that surface area. Another way of looking at this is imagine that you have a small sponge. You take that small sponge and you place it in an area that has got some red dye in it, okay? So let me change my color here. And when you place it in an area that has red dye, that red dye will soak up that entire sponge, okay? So it soaks up that entire sponge. If your cell, in this case, was larger, and you added the same volume of red dye, your red dye in this case might only just soak up the very edges. So what does that mean in terms of oxygen going in, oxygen going out? Well, what that means is if you are smaller, your oxygen can go in and go out and can be more efficient in a smaller cell. In a much larger cell, the cell is not as efficient. It can go in on the very edge and go out on the very edge. Yet the whole interior part of that larger cell is starved for oxygen. It's starved for carbon dioxide. So why are cells so small? Because they're much more efficient when they're small. They're much more efficient when they're small. So the question is, well, why aren't they infinitely small? Why aren't they infinitely small? Well, they're not infinitely small because of this diagram right up here. In this diagram up here, you can see all of the machinery that's inside the center of that cell. You've got the nucleus that you probably heard about when you very first took biology way back in the day. You've got the endoplasmic reticulum. You've got the mitochondria. You've got the cytoplasm. So you've got the machinery inside that cell that needs to drive that cell. And so that's why they're small, but they're not infinitely small because you need that machinery in order to drive that cell. So this is kind of just exactly what we just talked about. What limits the cell size? Well, what limits the cell size is actually that surface to volume ratio. That's what limits the cell size. And so again, this is looking at that larger cell that I just drew for you. Oxygen going in, oxygen going out, yet the center of that cell is starved for oxygen. This, what looks like a Rubik's Cube here, with all the little itty-bitty tiny cells in it. So, I don't know, 100 or so itty-bitty tiny cells. Each one of those independent tiny, itty-bitty tiny cells, there is a much larger volume there because each tiny cell has its own volume to it. So there's oxygen going in, oxygen going out in each one of those independent tiny little cells. Oxygen going in, oxygen going out, oxygen going in, oxygen going out. And so it's a much more efficient of a mechanism, if you will, if you are made up of all these itty-bitty tiny cells. Interestingly enough, the oxygen going in, oxygen going out actually moves across the surface in the process called diffusion. So diffusion means that there is a movement. Oops. There's a movement across the membrane without any energy. And interestingly enough, what we have is the movement across this kind of Rubik's cube situation down here. We have the movement across that cell membrane of oxygen, food, water, things like that, and then we get rid of the waste product, which is carbon dioxide. So this is looking again at that limit to that cell size. It says the metabolic requirements set that upper limit. In large cells, it's much more difficult to move that material inside and outside fast enough to support life. And so that's exactly what we have been just talking about. So what is this process? Well, the process that we just talked about in the previous slide, the process of moving across that cell membrane is diffusion. So diffusion is movement across a membrane without any energy. So we have oxygen down here that is diffusing across that membrane, amino acids that are diffusing across that membrane, and then carbon dioxide, for example, and ammonia that are the byproducts that are being released and going in the opposite direction. So we have items that are diffusing or moving across that membrane. And the smaller the cell, the easier it is for those products to move back and forth across that membrane. What we have here is a cell that is very, very large. And it's going to take probably energy to actually move those products across a cell that is that large. And so again, it's much easier to have much smaller cells so you can actually move products to and fro without any of that process that we call diffusion. So what we have is organisms that we call multicellular organisms. So you are a multicellular organism, your cat is a multicellular organism, your dog is a multicellular organism. So what we have is anything that is made up of more than one cell is a multicellular organism. So what challenges do multicellular organisms face? Well, there's quite a few that they face. They first have to have that ability to bathe their cells in different nutrients, and each cell has to have that ability to bring oxygen in and remove carbon dioxide and remove nitrogen, and that has to be a process that is done readily. And so you can see here, this is a multicellular organism, if you will, and oxygen is diffusing into that, amino acids are diffusing into that, and then the waste products of carbon dioxide have that ability to actually move out of those cells once the carbon dioxide levels or the nitrogen levels build up to a specific level, then they move out of those cells. So what we're going to do now is we are actually going to kind of do a tour de cell, if you will, and we're going to start looking at the characteristics of different cell types. And so what we know is that all cells are surrounded by a plasma membrane that has cytoplasm, which is kind of a semi-fluid substance that has a membrane, and those cells contain organelles. And we're actually going to take a look at those specific organelles. So before we get started with that, what I'd like to do is move forward and look and discuss that there are actually two different major types of cells that I want you to know about. And the first major type of cell is what we call a prokaryotic cell. And so number one is a prokaryotic cell. These prokaryotic cells are your cell types like bacteria is an example of a prokaryotic cell. And another type of prokaryotic cell, which I don't have listed on this slide, is something that we call which is my favorite, the archaea bacteria. And the archaea bacteria is the extremophile bacteria. And the extremophile bacteria, I think if I ever come back reincarnated as a bacteria, I'm going to be an archaea bacteria. The extremophile bacteria are the ones that can live around hot thermal vents, which are deep, deep down in the ocean, and spew out water that is almost like boiling water. Or if you ever go to see Old Faithful, where the water is really, really hot, you know, that is the archaea bacteria. Or these sulfur vents where they're spewing out sulfur water, the archaea bacteria can live there. There's some caves that have stalactites and stalagmites, and they've got some of the stalactites that are hanging from the surface of these caves actually have a sulfuric acid that's dripping from them, and lo and behold, that bacteria can actually live in that kind of sulfuric acid environment. So yeah, I'd step out of the box and I'd be one of those bacteria. So the prokaryotic bacteria are very, very simple types of bacteria, and the extremophile types of bacteria actually live inside that very simple form. They don't have any types of organelles inside of them, and that's kind of what puts them into that kind of prokaryotic or very simple type of bacteria. The other type of, let me see if I can get my pencil up here. The other type of major cells is number 2, is what we call the eukaryotic cells. And so the major 2 types of cells, one, prokaryotic cells, in which they lack organelles, they lack a nucleus, and number 2 is the eukaryotic cells, in which they have organelles and they have a nucleus, and they're more complex of cells. So if we were to take and kind of put all cells together, what do all cells have in common? So all prokaryotic cells and all eukaryotic cells, what do they all have in common? Well, all cells have in common a cell membrane that surrounds the outside of them. All cells have in common ribosomes, even though those ribosomes may look a little bit different and may be slightly different from one a cell type to another, they still have ribosomes, and all cells have DNA. So those are characteristics that all cells have in common. So this is looking at types of cells, again, that prokaryotic cell, a very, very simple cell, versus a eukaryotic cell, which is your much more complex cell, and looking at kind of the specific differences between the two. So your prokaryotic cells, and you can read through these on your own, are your more simplistic cells. The DNA is kind of coiled up into the center of that cell. Not too much, there's not too much to that cell. I'm going to show you a picture of it in just a second. And then your eukaryotic cells are your much more complex cells. They're the cells that you learn about in your basic biology courses, in which you've got a nucleolus, a nucleus. You have your basic animal cells, your basic plant cells. These are your much more complex cells in which you have membrane-bound organelles. And we're going to actually go through some of those membrane-bound organelles as today continues. So let's start out first by looking at what the shape of that prokaryotic cell, that very, very simple cell, looks like. So this again is that prokaryotic cell, again, the simple cell structure. So again, the bacteria, or that extremophile bacteria that lives in those extreme environments. As you can see, these cells lack a nucleus in the center. They have their DNA that is just literally kind of coiled here in the center. It's not surrounded by a membrane at all. It's just kind of hanging out loosely, kind of coiled around in the center. They've got some ribosomes, and so their ribosomes are kind of here, here, and here, just kind of hanging out. Kind of cool. They've got a cell wall, so they've got a cell wall that kind of comes around here. Ta-da! They've got some cool flagella, so nice for them. Whether they use them or not, who knows? But they've got those, so that's kind of neat. They've got these kind of what we call papillae that are up here. So not all, but most do. Perfect. So that's that basic structure of the prokaryotic cell. Not much to it. It also has this kind of characteristic capsule shape to it. Kind of looks like a little pill, if you will. So this is looking at the eukaryotic cells. And why is it that eukaryotic cells have got organelles? And the answer to that is go to the Container Store. And if you've ever gone to the Container Store, which, oh my gosh, I love the Container Store. You can find a container to put just about everything. Come to my house and you can actually open up one of my cabinets and see all these, organizing containers. It's fabulous, just fabulous. So if you delve inside the world of a eukaryotic cell, remember the eukaryotic cells are the more complex cells. These are your animal cells. Oops. And plant cells. So it seems reasonable if you were talking about animal cells, you're talking about plant cells, that if you kind of open up one, crack one open and look at the parts inside, it seems reasonable that it would be like the container store inside that animal cell or inside that plant cell. You would have to kind of compartmentalize the different factories or the different parts inside that cell, because you don't want to mix your different apparatuses that are going on. So if you've got a digestive enzyme, you don't want that loose because it's going to digest the whole cell away. So you want to put it in this little tiny container and have it do its digestive job inside its little container, hence inside its organelle. You want to have another compartment over here, so it's not, doing and, having an effect on another part of the cell. And so everything has to be partitioned off and placed into different compartments. What we have is everything is placed into different compartments. Let me see if I can. Let me see if I can stop this for a second. See if I can get that guy to go. Okay, so everything is placed into different compartments. We have sorry about that. So everything is placed into different compartments. We have things that are different pHs. We have different concentrations of lysosomes and digestive materials. And gosh darn it. Hopefully that person will go away. Anyways, let me see if I can get back to this. Let's just move on. I think you guys get that. Let's move on. So let's look at how the cell works. We know that the animal cell and we know that the plant cell has got different jobs. And so let's concentrate on this right here being the animal cell, and this right here being the plant cell. And the animal cell and the plant cell, again, are both eukaryotic cells, and each of them have got different functions. And so the functions of an animal cell, the functions of a plant cell, are as stated here, that they are to make proteins, they are to make energy such as ATP for daily life. So you can actually pack your backpack and drive your car and go from point A to point B. They are for growth and repair of cells, and they are so if you get a cut and your cut needs to heal, so hence growth and repair of cells, and to make more cells. So I just had a brand new nephew that was born about 6 days ago, and in those 6 days I've actually watched my little nephew grow, and I swear he is looking more alert, and his little eyes are tracking more, and so I've actually watched him grow, and it's been an amazing thing. So cells grow, cells repair, and cells renew. If you think about your own skin, you've got skin renewal that is happening on literally a daily basis. So that's kind of a renewal. All right, so let's kind of step back and let's take a tour to cell. We're going to go through some different components of the cells. I'm sure that each and every one of you have had the cell in probably high school or another type of basic biology class. And so I would hope that you guys already know the different parts of the cell. So we're going to kind of cruise through it here and give you guys just kind of some of the highlights of the different parts of the cell. So the tour to cell. So let's take a look at kind of the central point of the cell, and the central point of the cell is this right here, which is the nucleus. And this image isn't showing us the nucleolus, but there is the nucleolus, which is kind of a center part, which is like a little dot right inside the nucleus. And the nucleolus, again, is found within the nucleus, and all the chromosomes are actually kind of located within the nucleolus, and so they are contained within that nucleolus, and the chromosomes are actually producing ribosomal RNA to actually make ribosomes. And so that's kind of a neat thing that we actually don't think about. When we think of the nucleus itself, we think about the nucleus containing DNA, and nucleus kind of being kind of the control center, if you will, of the cell. So there's a kind of a running list here of different organelles that we are going to kind of highlight. So ribosomes, the endoplasmic reticulum, so we have smooth and we have rough, and one of my favorites is the Golgi apparatus and transport vesicles. So we're going to kind of look at all of these different vesicles and organelles, and something that I kind of want you to keep in mind is kind of the protein assembly line, and how is it that you actually make proteins. And I already kind of gave you a hint on how to make proteins, but this is our protein assembly line right here. And so you might kind of hear that in your future, how do you make a protein? And so this right here is your protein assembly line. So you might want to kind of remember this, and we'll talk about this as time marches forward. So if we think about the ribosomes, there's a couple of different places where the ribosomes are actually located. We've got free ribosomes, which are actually suspended in the cytoplasm, And so this is looking at free ribosomes that are actually in the cytoplasm, and so you can kind of see them dotted around here. We also have bound ribosomes, which are actually attached to the endoplasmic reticulum. And so I always think that these kind of look like, I don't know, pita pockets, but I also think that the Golgi apparatus looks like pita pockets. But you can see these little tiny blue dots here that, to me, kind of look like little little, I don't know, sesame seeds, if you will, that are kind of hung out here on this kind of pita pocket situation going on. And the job of a ribosome its function is to build a protein. So it says synthesize a protein, build a protein, same thing. So its job is to build a protein or synthesize a protein and then export it out of the cell. How does it export it out of the cell? We'll talk about that in a couple of minutes, but that's its job. The ribosome's its job is to build a protein, to create a protein, to synthesize a protein. We talked about the electron microscope before, and so this is kind of a cool transmission electron microscope image of those ribosomes. So this is kind of an up-close and personal picture of the rough endoplasmic reticulum. Again, down here in this image, we're looking at kind of the pita pocket image, if you will, and you can see the little dots on that. Those dots are the little tiny ribosomes, and I'm trying to put little dots on top of the dots, but you get what I'm talking about. Those are the little pita pockets. And what we have here is a protein is actually created up here in the nucleus, and that protein will come down here and will be actually synthesized in the ribosomes of the endoplasmic reticulum. It will actually be secreted into and packaged into a transport vesicle right here, and will be then packaged and moved on to the Golgi apparatus. And so this right here is the Golgi apparatus. And so this kind of pita pocket looking structure here is the Golgi apparatus. The rough endoplasmic reticulum, I always think of the rough endoplasmic reticulum is kind of being, oh, I don't know. So if this is kind of the membrane of the rough endoplasmic reticulum, and this is a ribosome, what we have is our messenger RNA kind of comes up and goes into that ribosome, and that, so this is our ribosome, and this is our membrane here of the rough endoplasmic reticulum, and our messenger RNA again comes up into here. It goes up into that ribosome, and it tells it to make a protein. And so the rough endoplasmic reticulum is a site where we'll actually make those proteins and then package it on. So I'm actually going to skip over this and move on to the Golgi apparatus, because once the protein is made from the rough endoplasmic reticulum, from the rough ER, what it's actually done is it actually moves on to the Golgi apparatus. And the Golgi apparatus, what I'd like you to do is think of the UPS, United Parcel individuals, the guys that come in, might be girls too, that come in the brown trucks. And the dogs in my neighborhood love the United Parcel guys that come to my house and bring me packages. So at the United Parcel individuals, what they do is they receive packages, they modify those packages, they sort those packages, they ship those packages to your house. And so that's exactly what the Golgi apparatus does. It receives that transport vesicle with the protein in it from the rough endoplasmic reticulum, and so what it does is it'll that transport vesicle will come from the rough endoplasmic reticulum, and it will arrive at the Golgi apparatus. So the Golgi apparatus receives that transport vesicle with the protein in it. It will then modify, sort, and then ship that that package. So let's take a look at that. So when you think of the Golgi apparatus, think of the United Parcel guys. So this is looking at the rough endoplasmic reticulum up here. Let me change that color to red. This is looking at the rough endoplasmic reticulum up here. Remember, that's what's creating the protein. It places it into a transport vesicle, and then the Golgi apparatus receives that transport vesicle. Okay, so again, this kind of tan color business down here is the Golgi apparatus. It receives a transport vesicle. So here's the transport vesicle, and it shows up at the Golgi apparatus. When it shows up at the Golgi apparatus, it actually, the membrane of the transport vesicle fuses with the membrane of the Golgi apparatus. When those two membranes fuse together, what happens is the transport vesicle opens up. And when it opens up, it releases its contents into the Golgi apparatus. And so it releases the protein contents into the Golgi apparatus. So what the Golgi apparatus does with those contents is it sorts those protein chemical contents according to certain destinations. And so some are going to go here, some will go there. And then what it does is it'll actually repackage. So let me get a different color. Let me get purple. So it'll repackage those contents into different transport vesicle packages, and it'll ship it out to other places in the cell. And so it'll move across that Golgi apparatus. It'll reship the contents into different vesicles. And so this vesicle here might have, let's say, all amino acids, and it will go to this part of the cell over here. This transport vesicle here might have all digestive enzymes, and once it goes through the Golgi apparatus, it will be shipped out and it will go off to this side of the cell over here. Okay, so that's the Golgi apparatus. It receives the transport vesicle from the endoplasmic reticulum. Its membrane fuses with the Golgi apparatus. When the membrane fuses with the Golgi apparatus, it opens up and releases its content. Its content, then the proteins, then are shipped and sorted by the Golgi apparatus, and then repackaged into different packages, i.e. transport vesicles, and then they move across the Golgi apparatus membrane, and then they're repackaged into different vesicles, transport vesicles, and then those transport vesicles are shipped out the opposite side of the Golgi apparatus into different parts of the cell. And so that's where they're transported here to different parts of the cell, and then transported here to different parts of the cell. And this right here puts that whole story together. And so what we have here is the nucleus of the cell. Here we have the nuclear membrane shown in green. In orange, we have the rough endoplasmic reticulum, and then the purple dots are the ribosomes. The ribosomes create the protein shown here. This is, oh, of course, that's purple. Shown the proteins shown here, these purple dots are ribosomes. And what it does is once those proteins are made, those proteins are packaged into a transport vesicle. And so this right here is your transport vesicle with the protein inside. Your transport vesicle then moves in the cytoplasm and moves from the rough endoplasmic reticulum to the Golgi apparatus. And this right here is the Golgi apparatus. This is where the transport vesicle binds to one side of that Golgi apparatus. So its membrane binds with the membrane of the Golgi apparatus. It opens up, it releases the protein contents, and the Golgi apparatus sorts those protein contents according to the contents. Once it determines the contents inside that vesicle, it will then repackage the contents into different vesicles, and once it repackages them into different vesicles, so now we have vesicle 1 up at the top, vesicle 2 at the bottom, it then will move them across the membrane and it will ship them in these vesicles to other places. Okay, so that is how proteins are made and how proteins move around in the cell. So the only piece of information that I skipped over is the smooth endoplasmic reticulum. So the smooth endoplasmic reticulum, and I went back to slide 16, I think, it looks like pieces of spaghetti, if you will. So if you look at this image right here, you can see that it kind of looks like pieces of spaghetti that are kind of cut off. And then this is kind of an aerial view of it. So you're looking down on those pieces of spaghetti. So smooth endoplasmic reticulum lacks those ribosomes, hence it's called smooth. And smooth endoplasmic reticulum, it's known for having many metabolic processes. And so its job is to synthesize lipids, so oils and phospholipids, which is involved in kind of that membrane of the cell. It's involved in creating steroids and sex steroids, so testosterone and estrogen that you might be aware of. And it's also involved in a process called hydrolysis. And so the hydrolysis of glycogen into glucose. And so remember, hydrolysis means to add water to something. And when you add water to something, you actually break it down. And so you detoxifying the liver is to break down detoxifying drugs in the liver, to break down those drugs that you take. So whenever you take a over-the-counter drug, whether that is an aspirin or a Tylenol, whether that is a prescription drug, whenever you take a drug into your system, your liver literally has to break that drug down in order for your body to be able to utilize that drug. And whenever you consume alcohol or some type of painkiller, whether that be a barbiturate or what have you, your body has to break that alcohol down or break that painkiller or barbiturate down. And so that actually takes place in the liver and takes place through the smooth endoplasmic reticulum. All right, you guys, so make sure that you know the main organelles in the eukaryotic cell. It's kind of a laundry list of the different organelles. Make sure you know a little bit about what each one does. And I've had to cut this lecture up into part 1 and part 2. So part 1 is the biggest one. Part 2 will just be a few slides, nothing major, and I will finish that up in part 2. All right, you guys are doing great. I'm proud of you. I'll talk to you later.